Hello student. So now again for some time we have to continue with the online teaching method. So we discussed about the our last topic, that is a blood group and physiological basis of blood transfusion. So this is very important topic, blood group. So it is very important for your theory as well as practical, as well as all your clinical side. This topic is very important. So as you know, there are different type of blood group systems are there. In that main blood group system are ABO blood group and Rh blood group. Okay, so ABO blood group, that is depend on the presence of the antigen on the RBC membrane. So human red cell contain numerous surface structures. They are recognized as an antigen by the immune system. These surface structures, that is blood group antigens, based on the presence of the antigen on the RBC membrane, corresponding antibodies are absent in the plasma of that individual. This forms the physiological basis of blood groups and principles of blood transfusion. So blood group antigens, they are known as agglutinogens, and antibodies against these agglutinogens are known as agglutinins. And the reaction between the agglutinogens and agglutinins result in the agglutination of RBC. So there are more than 30 known blood group system present over in the blood containing different 400 antigen. Many of them have cold antibodies. They do not react at the body temperature. Only few of them are immunological active. So for our ABO and Rh blood group is important for us. Other major blood group are also MNS, Lewis, Duffy, Cal, P and Lutheran system. But in our syllabus we talk about this ABO and Rh blood group system. So this table that showing the different blood group system and gene involved in this blood group system and total antigen. So in ABO blood group there are 4 antigen, Rh blood group there are 45 antigens are there, and other blood groups are given. So this question is asked in Viva, what are the use of blood group? So blood group has many use. So RBCs are less active cell, contain mainly hemoglobins. So they play important role due to presence of the antigen on their surface. So characterization of the RBC antigen antibodies form the basis of compatibility testing for blood transfusion. So when the blood group of individual is known, in the emergency situation, blood can be transfused with the suitable blood. So therefore ABO and Rh blood group are usually displayed in the identity cards of individual. The blood group antigens have provided the scientific basis for understanding the hemolytic disease of the newborn and autoimmune hemolytic anemia. Blood group antigen plays an important role in susceptible to infection by malarial parasite, bacteria and viruses. Antigens also associate clinical disorders, absence associated with specific dysfunction, inherited and acquired disease. So medical legal expert take the help of characterization of the RBC antigen antibody in establishing the identity of father in case of disputed paternity. Specific blood group have been reported to be associated with the human behavior. They also play important role in personality of individual. So these are all uses of blood group system, mainly paternity dispute, medical legal cases, susceptible to infection, clinical disorder, blood transfusion, human behavior, complete test for blood transfusion, and scientific basis for underlying hemolytic disease of newborn and autoimmune hemolytic anemias. Now discuss in detail about ABO blood group system. So ABO blood group system was the first blood group system that is known in 1901. So this is the most important system in the transfusion medicine because ABO agglutinogens are highly antigenic. So it is most significant of blood group system because of the natural occurrence of the A and B antibodies in the plasma of the individuals who lack corresponding antigen on their RBC. So in addition, transfusion of incompatible ABO blood group leads to serious transfusion reaction. So in ABO system there are two antigens, they are the antigen A and antigen B. A antigen has two subtypes, A and A1. Genes for antigen for ABO are located on chromosome 9. Okay. So these ABO antigens present on membrane of the RBC. They also found in the salivary gland, testes, lungs, kidney and pancreas. They are secreted in the minute quantity in saliva, pancreatic secretion and seminal fluid. Some individuals like blood group O secrete blood group H antigen in the saliva and other body fluids. They are known as H secretors. So ABO antigens are present since fetal life, but they first appear at 6 weeks of intrauterine life. At birth, the concentration is about 1/5 of the concentration in adults. Concentration slowly rises to reach a significant level at puberty and reaches maximum at 15 to 17 years of life. Therefore, for collection of blood for blood transfusion, the age of donor should be minimum of 15 years. So donors age should be above 15 years. So concentration of ABO antigens rise gradually and reaches its significant level at 15 to 17 years. Therefore, for collection of blood for blood transfusion, the age of donor should be minimum of 15 years. So based on the presence of antigen and absence of antigen on the RBC, the blood group are described as a group A blood group in that antigen A is present, group B blood group B antigen B is present, group AB both A and B antigen present, group O neither A nor B antigen is present. Okay. So if we see distribution in Indian population, ABO blood group, so A blood group having 27%, B blood group having 31%, AB blood group having 8% and O blood group having 34%. In the A blood group, the A1 is 75%, A2 is 25%. So what is the role of H gene and H antigen? So the expression of the A and B antigen are dependent on the presence of H gene. Most of the individuals are homozygous for the H gene. So sequence of events in the formation of A and B red cell antigen is A and B red cell antigens are glycoproteins and their formation start with the basic precursor substance. Basic precursor substance converted to H substance under the influence of H gene. H substance partially converted under the influence of A and B genes into A and B antigens. Some of the H substance remain unconverted. Since the O group do not have A and B genes, neither A or B antigen is formed. So these have only H substance. A, B and H antigen may be detected in the saliva and other body fluids. Such individuals are called secretors and remaining without called non-secretors. So that is about H substance and H gene. So basic precursor substance that form H substance, then no A and B gene blood group, B gene B antigen, A gene A antigen. So that is in the presence of N-acetyl galactosamine transferase and D-galactosyl transferase. Basic precursor substance contain H substance by L-fucosyl transferase. So this table is important for your MCQ. This enzyme can be asked in MCQ for your entrance test. So this is Bombay blood group. This is also asked as an objective question and also in Viva. So Bombay blood group that is discovered in 1952 by Pinde, Bhatia and Deshpande. So these individuals of Bombay blood group that lack the H gene, and therefore the basic precursor substance cannot be converted into H substance. So this then results in the failure to form the A and B gene, B antigen. So when their blood sample is tested for routine AB grouping, they will be labeled as a blood group O. However, their serum contain anti-A and anti-B and anti-H antibody. So these individuals should be transfused with only Bombay blood group. So ABO agglutinins. In ABO system, the agglutinins are two types, anti-A alpha and anti-B. The alpha agglutinin has two subtypes. Okay, so alpha proper and alpha 1. So these antibodies are found in the plasma. Specific antibodies are formed with a particular antigen where they are exposed to that antigen. Blood group antibodies are naturally occurring antibodies. They are formed without prior exposure to the antigens and present in the blood of individuals in whom the respective antigens are present. Anti-A and anti-B agglutinins not formed during fetal life and absent at birth. They appear in the second week of neonatal life and increase very slowly in concentration to attain the peak level at about 10 years of life. It has been proposed that intestinal bacteria and food contain antigens similar to that of blood group agglutinogens. Therefore, when babies start eating and food gets absorbed from intestine, antigens are absorbed into blood. As these antigens are recognized as non-self antigen by body's immune system, they stimulate the antibody production. These antigens are present at very low concentration. Antibody against them are produced very slowly. Therefore, antigen-antibody reaction does not occur. So, antigens are removed by phagocytic cell. ABO incompatibility does not cause hemolytic disease of newborn. ABO antigens are also expressed in the other tissue. Therefore, ABO compatibility is a significant consideration in organ transplantation. However, in the newborns, the ABO incompatibility rarely causes clinical hemolytic disease because antibodies against the A and B are predominantly IgM and they do not cross the placenta during the fetal life. So, an antigen A and B are not well developed at the birth. So, ABO incompatibility does not cause the hemolytic disease of newborn. This can be asked as a explained in the 1 or 2 sentence or objective question. So, A and B antibodies are globulins and belong to IgM category. They do not cross the placenta. They are known as cold antibodies and they function efficiently at low temperature between 5 degree to 20 degree Celsius. Antibodies are directed against the antigen. The individuals with the blood group A antigen on the RBC membrane do not have the anti-A antibody and possess anti-B antibody in the plasma. This means this if person having the blood group A antigen, then in the plasma the anti-B antibody is there, and in blood person with the blood group B having anti-A antibody in the plasma. So individual with the blood group AB do not have any antibody. Individuals with the blood group O have both anti-A and anti-B antibody in the plasma. So this fact was first noted and discovered by Karl Landsteiner in 1900, and this theory is popular by Landsteiner law. Karl Landsteiner discovered this agglutinin in the human blood, leading to recognition of human blood groups and success of blood transfusion. Principle of presence or absence of antigen and antibody in the RBC and plasma in various blood group is known as Landsteiner law. He received the Nobel Prize in Physiology or Medicine in 1930. So this you have to remember, Landsteiner law. It can be asked in the viva or in the short question. What is Landsteiner law? So if a particular agglutinogen antigen is present on the RBC membrane of individual, then corresponding agglutinin means antibody must be absent in this plasma. Conversely, if the agglutinogen is absent in the RBC, means antigen is absent in the RBC, corresponding agglutinin antibody must be present in the plasma. This law holds good for the ABO system. The second part of the law does not apply for Rh blood group and many other blood group system, because there are no naturally occurring antibodies in this Rh system and other system. So this is about the Landsteiner law. In ABO system, the blood group A has antigen A on the RBC membrane and anti-B antibody in the plasma. So blood group B has antigen B on the RBC membrane and anti-A antibody in the plasma. Blood group AB has both antigen A and B on the RBC membrane and no antibody in the plasma. And blood group O has no antigen on the RBC membrane and both the antibody A and B in the plasma. Okay, so understand. So this table showing the occurrence of antigens and antibody in the ABO system. So if you see in the blood group, person having blood group A having antigen A and antibody B. B blood group having antigen B and antibody A. AB blood group having A and B antigen and no antibody, and O blood group having no antigen and antibody anti-A and anti-B. Blood grouping is performed by suspending the RBC of the person's blood group to be checked in the anti-A and anti-B anti-C. They are commercially available. If agglutination is present, means clumping of RBC, if checked under microscope, then RBC contain that particular antigen. So this figure that's showing the practical of blood grouping. So in the first figure you can see agglutination occur in the anti-A, means the blood group is A. In second slide you can see agglutination occur in the anti-B, so blood group B. In third, there is no agglutination in anti-A and anti-B, so blood group is O. And in the last is agglutination in both A and B, so blood group is AB. So in this way you have to find the agglutination and how will you determine the blood group on the basis of agglutination. Then inheritance of ABO blood group. Blood groups are genetically determined. Normally the presence of blood group antigen is dominant characteristic. So antigen is present in the phenotype irrespective of genotype. So there are three type of genes are there, O, A and B. So persons have any of these six possible genotype, O, A, OB, then AA, AB and AA AB and BB. Gene O is not important, so respective phenotype may be A or B or AB or O. So if blood group of one parent is A, other parent is B, so there is possibility of child having any of the four blood group of ABO system. Father is A, mother is B, father genotype AO, mother genotype BO, so gametes AOBO. So genotype is ABAO, then OBOO. So the AB, A, B or O. So A blood group having genotype AA and AO, B blood group is BB, BO, AB having AB and O having O. So knowledge of inheritance of blood group is useful to solve the cases of disputed maternity. Inheritance of blood group is useful to solve the cases of disputed paternity. So when the blood group of the child and mother is known, so it can be told whether man in and suspicious could have been the father of the child or not. However, blood group can never prove that particular man is the father of particular child. This is because the blood group of the man can be one out of several other possible men. So therefore determination of blood group gene only helps to identify, not confirms the identity. MNS system is very useful in this regard. Disputed paternity. After discussing the ABO blood group system, we discuss about the RH blood group system. So RH rhesus system is the second most important blood group system in transfusion medicine. This was first described in rhesus monkey in 1940 by Landsteiner and Weiner, so it is known as RH system. First it was discovered that injection of red cells of rhesus monkey into rabbit results in the development of antibody against the rhesus RBC. Later it was found that rabbit serum containing anti-rhesus antibody agglutinated the human red cell. This led to discovery of RH system in the human. In RH system there are 6 antigens and there are no naturally occurring antibody. These antigens are C, D, E, capital C, D, E and small c, D and E. Out of 6 antigen, immunological most active is a D antigen. Thus in the RH system there are 2 blood groups, RH positive and RH negative. The gene for RH antigen is RHD and RHC genes, they are located on the chromosome 1. The antigens of RH system participate in the cation transport and membrane integrity of RBC. The RH antigens are membrane proteins found in the RBC membrane, not in the other tissue. RH antigens inherited as a dominant gene. In Indian population, the 98% of RH positive and only 2 to 5% RH negative. If RH positive individuals may have homozygous or heterozygous genotype, 60% of RH positive have DD genotype and 40% have capital D genotype. The genotype of RH negative person is small d. RH negative person is small d. If the one of the parent is homozygous positive and other is homozygous negative, all offspring will be heterozygous positive. Similarly, if the father and mother are homozygous negative, all offspring will be homozygous negative. And one of the parent is heterozygous positive and other is homozygous negative, and 50% of offspring will be heterozygous positive and 50% is homozygous negative. So this is the inheritance of Rh antigen. Rh antibody is anti-D antibody, and this antibody produced only when Rh negative individual receives the Rh positive blood. This antibody is developed very slowly in the first encounter, but form rapidly following subsequent encounters. Rh antibodies IgG type which can cross placental barrier. If antibodies are present in the mother's blood, can be transferred to fetus. Rh antibody is base reactive at body temperature and therefore designated as mom antibody. So ABO antibody is cold and Rh antibody warm. Rh incompatibility occurs when Rh negative individual receives the Rh positive blood. Normally when the Rh negative person receives the Rh positive blood, there is no immediate reaction, as Rh negative individual does not normally has anti-Rh antibody. The donor's red cell induce the immune response in the recipient to synthesize the anti-Rh antibody, which takes about 2 to 4 months to reach the significant titer. However, by that time donor's RBC dies a natural death within 120 days. So anti-Rh antibody cannot produce any harm to the recipient's RBC because Rh negative recipient red cell contain no Rh antigens. But if the same Rh negative persons who has already received Rh positive blood before and receives second Rh positive blood later, anti-Rh antibodies are synthesized large amount immediately by the memory cell. This antibody reacts against the donor cell and causes reaction of mismatch transfusion. Thus, Rh negative individual can safely receive Rh positive blood once in the lifetime. Similar Rh incompatibility occurs in pregnancy when Rh negative mothers bears Rh positive fetus, which leads to erythroblastosis fetalis. So phototherapy is needed for neonatal jaundice. Bilirubin in excess is toxic to body, may prone neurotoxicity. On exposure to the light, bilirubin becomes non-toxic. So this is a physiological basis of phototherapy. Bilirubin exposure to light undergoes rupture and causes isomerization and photooxidation. These changes in bilirubin makes less toxic and less lipophilic products. They are extracted without hepatic conjugation. Therefore, phototherapy is primary treatment for unconjugated hyperbilirubinemia in the newborns. Neonatal jaundice is similarly hemolytic and unconjugated. So this can be asked as an objective question. What is erythroblastosis fetalis and Rh incompatibility? So erythroblastosis fetalis is the hemolytic disease of newborn which occurs due to Rh incompatibility when Rh negative mother carries Rh positive fetus during pregnancy. Usually there is no reaction in the first pregnancy, but at the time of delivery during placental separation, small amount of fetal blood leaks into maternal circulation. So this induces the formation of anti-Rh antibodies in the mother. So in subsequent pregnancy, this anti-Rh agglutinates from the mother, which is predominantly IgG type, crosses the placenta to enter the fetal circulation and causes hemolysis because antigen is present in the fetus and antibodies are come from the mother blood, so it causes anti-antibody reaction, so hemolysis of RBC occurs. In third and subsequent pregnancy, the degree of hemolysis becomes very severe. So, clinical features are mainly due to hemolysis. So what is the erythroplastosis fetalis? You understand? This mother is Rh negative and it carries fetus Rh positive. So no reaction at the first pregnancy, but at the time of delivery during placental separation, small amount of fetal blood mixed with the mother blood. So when the blood mix occurs, so fetal antigen enter into mother blood. So that antigen is foreign to mother blood. So in mother there is production of anti-Rh agglutinase antibody in the mother. So this antibody remains in the mother for a very long time. When the second pregnancy occurs, when the same time mixing of blood of fetus and mother occurs, This time the presence of antibody in the mother and antigen that comes from the fetus, it produces anti-antibody reaction and hemolysis occur. So features of this erythroblastosis vitalis mainly due to hemolysis. So hemolysis leads to anemia, extramedullary erythropoiesis, hematopoiesis and neonatal hyperbilirubinemia. If hemolysis is severe, the fetus may die in the uterus. If the born alive, they may have the following features. Anemia occurs due to degree of hemolysis. Hemolytic jaundice that occur due to hemolysis. Serum bilirubin may be more than 25 mg percent in severe cases. Generalized edema. Edema occurs in the whole body due to anemia and hypoproteinemia. This is called hydrops vitalis. Kernicterus, that is more hemolysis. More hemolysis, there is release of more hemoglobin. So from breakdown of hemoglobin, bilirubin is release. So this more amount of bilirubin, it crosses the blood-brain barrier and it's deposited in the basal ganglia, that is part of central nervous system. So this kernicterus is a neurological syndrome with major defects that occur due to deposition of bilirubin in the basal ganglia. So hyperbilirubinemia occurs to hemolysis. Basal ganglia have more affinity for bilirubin, so bile pigments cannot cross the blood-brain barrier. So hemolysis in adults does not produce kernicterus. Because in the fetus, blood-brain barrier is not fully developed in infants and children, so in them bilirubin enters in the brain and get deposited in basal ganglia. Therefore, hemolysis in the infancy and early childhood causes kernicterus. Dysfunction mainly manifests in the form of the motor dysfunction. Extramedullary hematopoiesis. Due to severe anemia, extramedullary hematopoiesis occurs, for which erythroblasts are released into the blood. Erythroblasts are seen in the plenty in the peripheral smear, and this is called this erythroblastosis vitalis because there are plenty of erythroblasts in the circulation. So treatment of erythroblastosis vitalis, so 50% of fetus and newborns with erythroblastosis vitalis have mild symptoms, mild hemolysis and do not require treatment. In severe cases, the major modalities of treatment are intrauterine fetal transfusion, exchange transfusion and phototherapy. What is exchange transfusion? If disease diagnosed in fetus and found to be severe, treatment is intrauterine fetal transfusion. Fetal transfusion is carried by intravitreal route, which has replaced the direct intravascular fetal transfusion. Exchange transfusion treatment of newborns with severe anemia, jaundice and hydrops. Exchange transfusion after birth. Exchange transfusion removes the sensitive RBC, bilirubin and maternal antibody from the plasma. Double volume exchange transfusion 2 into 80 ml per kilo replaces 90% of infant's blood volume with antigen negative reaction. Blood chosen for exchange should be ABO negative, Rh negative. Its cross match against mother's blood. Intensive phototherapy is very effective in reducing serum bilirubin level. Prevention. Erythroblastosis vitalis is prevented by administering single dose of anti-Rh antibodies in the form of Rh immunoglobulin during the postpartum period following first delivery. The disease also prevented by passive immunization of mother with a small dose of Rh immunoglobulin during pregnancy. Other blood group system, MNS blood group system described by 1927. This system has 3 antigen, AM and NS. Antigen expressed on the RBC and epithelial cell of renal capillaries. MNS antigens are major contributor for negative charge on the RBC membrane. Common phenotypes MNS. System helpful for solving patented dispute. This MNS group is useful for anthropological and genetic studies. H system, the common antigen is H antigen. Expression of antigen depends on expression of the ABO groups. Blood group O having H antigen in the plasma, saliva for OH group. Gene for H antigen is FUT1 located on the chromosome 19. H antigen is present in the plasma, most body secretion like saliva, sweat, semen, tissue, epithelial cell. So they are known as secretors. Liver system described in 1946. two antigens, Li and Lep, also present in the body secretion and limb. Gene for antigen Li is located on the chromosome 19. Antigen for Lewis systems have a receptor for Helicobacter pylori. Antibodies are IgM type. They do not cause the placenta, do not cause the hemolytic disease of newborn. Lee system is there. Which are the disease associated with the blood group? So in a person having blood group A, is most chances of carcinoma of stomach, colon, ovary, uterus, ITP, colon thrombosis, pernicious anemia. Blood group B having chances of Bacillus infection, gonorrhea, UTI. Blood group O having the peptic ulcer, rheumatoid arthritis, typhoid fever, bulbariosis. Blood group RH negative O2 hemolytic anemia. Okay. So this is all about the blood group system. The next topic is blood transfusion, that is discussed on the next lecture. Okay.